Hello, Madan.
हजुर सर सुरु गर्दा हुन्छ सर
ओके हुन्छ हुन्छ
हजुर मदन खनाल IVSA RAMPUR 21-22 कमिटीको सेक्रेटरी र आजको यस वेबिनार सेसनमा म सम्पूर्णलाई स्वागत गर्न चाहन्छु। आजको यस वेबिनार सेसनको टपिक रहेको छ पोल्ट्री डिजिज डायग्नोस्टिक फेसिलिटी एन्ड इट्स फ्युचर पर्सपेक्टिभ र आजको यस हाम्रो वेबिनार सेसनको रिसोर्स पर्सन हुनुहुन्छ डाक्टर नवराज श्रेष्ठ। उहाँ अहिले सेन्ट्रल भेटनरी ल्याबोरेटरी त्रिपुरेश्वर काठमाडौँमा भेटनरी अफिसरको रूपमा कार्यरत हुनुहुन्छ र उहाँ मुख्यतः पोल्ट्रीमा प्र्याक्टिस गरिरहनु भएको छ र उहाँको स्पेसिस अफ इन्ट्रेस्ट भनेको स्पेसल्ली ब्रोइलर र आजको उहाँको प्रेजेन्टेसन अवधिभर सबैले आफ्नो माइक्रोफोन म्युट गरेर सहयोग गरिदिनु होला र यदि कुनै क्वेरिजहरू छन् भने च्याट बक्समा राखिदिनु होला अथवा उहाँको प्रेजेन्टेसन सकिसकेपछि आफ्नो माइक्रोफोन अनम्युट गरेर पनि सुन्न सक्नुहुनेछ। र अब म कुनै पनि ढिला नगरिकन सरलाई आफ्नो स्क्रिन सेयर गरेर प्रेजेन्टेसन सुरु गरिदिनु हुनको लागि अनुरोध गर्दछु। धन्यवाद।
ओके धन्यवाद मदन भाइ। यो के नेपाल हामी यहाँ इन्टरेस्टेड पार्टिसिपेन्ट पनि छौँ हो। मैले इङ्ग्लिसमै नेपाली इङ्ग्लिस के अरे नेपालीमा बोल्दा हुन्छ कि इङ्ग्लिसमा बोल्नुपर्छ मदनजी?
सर मैले पनि इन्टरनेसनल पार्टिसिपेन्टहरु देखिरहेको छु। आई थिंक इंग्लिश वुड बी बेटर सर।
Okay, thank you so much Madan Khanal ji. Okay, hello and good afternoon everyone. As introduced by Madan Khanal, my name is Dr. Nabaraj Shrestha. I am a veterinary officer working at Central Veterinary Lab, Tripureshwor, Kathmandu. Central Veterinary Laboratory is the only lab, it's a national reference laboratory for Nepal. So I'm working there for 2 years. So today's topic is on the poultry disease diagnostic facility and its future perspective. So in this presentation, I'll try my best to give the knowledge on the poultry disease diagnostic facility here in Nepal and its future plan, what we can do in the future for the poultry disease diagnosis. So Madanji, can I share my presentation?
Yes, sir, you can.
Excuse me, sir. Just before you share your, we have a queries from Vet Nepal, and he or she can live our presentation
yeah
session.
I think there will be no problem. I think yeah we can share this presentation in the public through Facebook also, but the only thing is that it is in the English, so I hope hello, am I audible?
Yes, sir.
Okay, so I think we can give, yeah, we can allow Vet Nepal to publish it in the public site. Okay, shall I start?
Yes, sir. Yes.
Okay. Okay, this is the topic for today: poultry disease diagnostic facility and its future perspective. Today's objective is to know about the history and development of poultry disease diagnostic facility in Nepal. So how the facility in Nepal regarding the poultry disease diagnostic is going on, and what are the diagnostic approaches that we are following to diagnose the poultry diseases, and also what are the modern and automated diagnostic equipments that we are having in the system. So these are the objectives that we will achieve after this session. I'm getting disturbed with the this waiting room. I think Madanji, shall we can we do that so that the participants can be allowed automatically? Because if we uncheck the this waiting room, then I think it will be easy for us.
Okay, sir.
Okay. Okay, so before we move into the presentation, I would like to share some limitation of this presentation. So here are basically two limitations, that we will not describe about the procedures and techniques that how we diagnose the disease. We just describe what are the facilities we are having here in Nepal regarding the poultry disease diagnosis. So we will not be discussing regarding the procedures and techniques. And here in the presentation, I have not prepared the slides regarding the facilities that are available in the academic institution and private sectors, because poultry disease diagnosis has been done by many academic institutions and private sectors. So here the information are basically from the Central Veterinary Lab and the other laboratories that comes under the Department of Livestock Services. So these are the limitations. I'll move to the topic now. We all know that the poultry sector has major 4 pillars, and those pillars are the poultry management, poultry health, poultry nutrition, and market. So today we will be just discussing about the poultry health pillars, and among the poultry health, we will be just discussing on the disease diagnostic facility only. And here you can see a few diseases of poultry that has been listed by OIE. I have listed this disease here because these are the disease of importance. So you can see here list A disease by OIE. There are 2 listed diseases, and list B diseases here are almost around 14 diseases that has been listed by OIE. These are the poultry diseases, and let me inform you all that there are total 117 diseases listed by OIE. And here in the left bottom side, you can see the notifiable diseases. There are total 24 notifiable diseases for Nepal, and among those 24 diseases, these 5 diseases are belongs to poultry diseases. So we all know that notifiable diseases means these are the diseases if we see in the field, then these are to be notified to the Department of Livestock Services, and these diseases will be notified to the OIE headquarters. So now here there are 2 types of diagnostic organization in Nepal. One is government and the other is private sector. So in government sector, there are a few veterinary laboratories, for example, CVL, Central Veterinary Lab, National Avian Laboratory, and veterinary laboratories you can see here. And along this side, there is also NARC, Nepal Agricultural Research Council. Though this council main objective is to do the research regarding the animal health. However, it can it is diagnosing, it is getting involved in the diagnosis of poultry diseases also. So that's why I have specified NARC as a diagnostic organization also. And there is Agriculture and Forestry University and Tripureshwor University. In this university also there is a teaching hospital which is engaged in diagnosing the poultry diseases. And here in the right part, you can see the VHL SEC. In fact, what I thought before this presentation was I will be just presenting for the Nepalese participants. That's why I have just specified its acronyms. So I'm really sorry to the international participants because these are the organizations and their acronyms short from. So I will describe it as far as possible so that I can make clear to you everyone. So VHL SEC means Veterinary Hospital Livestock Service Expert Center. These are the hospitals which is giving their services in the district level, and till now there are 59 VHL SEC. And let me inform you, all the international participants, that in Nepal there are 77 districts, and out of 77 there are only 59 veterinary hospitals at the district level. So in every veterinary hospital livestock service expert center, there are basic laboratories that has that are engaged in the diagnosis of poultry. And here in the private sector, you can see some of the names that HECAST and NPI. HECAST and NPI both are the private organization as they are affiliated to the Provincial University, and they do also have teaching hospitals, and they are providing the poultry disease diagnosis facility from their teaching hospitals. And there are many schools which are affiliated to CTVT and NEV. They are also servicing through their institutions, and similarly there are many I think there are no any data regarding the number of hospitals and clinics throughout the country. However, there are more than 50 clinics and hospitals in the country. So these are the institutions that are providing their service for the poultry disease diagnosis. So here is some history and development of veterinary laboratories here in Nepal. I have just tried to give more information. So in 1996 BS, I already told you that as I was not clearly informed regarding the international participants. So this PS is a Nepali calendar. So during the 1996 BS, the first veterinary dispensary hospital was established in the Tripureshwor, Kathmandu. And after that, in 2014 BS, 10 veterinary hospitals were established. And in 2049 BS, there was establishment of veterinary laboratories, one in each development regions. And since 2006, we are doing, PCR, that is the conventional PCR, and it is only available. It was only available at CVL Central Veterinary Lab, and it was before the outbreak of bird flu. And when there was a bird flu outbreak during the 2009 January, then there CVL purchased or started the RT-PCR for the diagnosis of bird flu. So in 2019, the Central Veterinary Lab has a diagnostic capacity of some automated system. I will later on describe this automated system. In 2019, we have Central Veterinary Lab has BD4X. It is one of the automated system. It was provided by NLSIP, Nepal Livestock Sector Innovation Project. And in 2020, Central Veterinary Lab has been supported by Fleming Fund with MALDI-TOF and Vitek 2. So regarding this automated system, I will describe later on. So in Nepal, we have BSL2 or BSL2 plus lab. As we all know that there are 4 types of biosafety lab, and out of 4 biosafety lab, our Central Veterinary Lab is a BSL2 plus lab. In fact, there is no any plus laboratories. However, Central Veterinary Lab is regarded as a BSL2 plus lab because it has equipments and it has a facility better than BSL2 lab, but it does not meet the requirement of BSL2, BSL3. So that's why Central Veterinary Lab is regarded as a BSL2 plus lab. And other veterinary labs, there are five veterinary labs in different regions. So these veterinary labs are regarded as a BSL2 lab. So these are the biosafety lab we are having in Nepal. And we do not have any BSL3 lab and BSL4 labs. Okay. So what are the diagnostic methods and facilities practiced in Nepal? Here are those. Okay, so I have, though this is not the facility, I mean, this is not a diagnostic facility. However, we have been practicing that history and clinical science are also a basis of clinical diagnosis of poultry diseases. That's why I have kept some of the information here in the presentation. As I already informed you that this is not a laboratory diagnosis. However, these are the basis for the disease diagnosis. So first thing is the history of disease. As you all know that history plays a major role in the disease diagnosis. For example, the history regarding feed storage, stress, vaccination, medicines, disease outbreak, and biosecurity. So this history, such type of history gives basic information for the disease diagnosis. And similarly, the type of birds also gives basic information for the disease diagnosis. For example, if you diagnose a disease, if you see the lesion of histomoniosis, if you see the lesion of histomoniosis in the turkey, then you are more likely to diagnose the disease. Your sensitivity of diagnosing this disease is very high, because histomoniosis is a disease of a turkey. Similarly, dog viral hepatitis, it is a disease of dogs. So the lesion and the type of birds is, there is some kind of correlation. So similarly, the age of birds also gives some information regarding the disease. For example, if there is a tumor in let's say 1 or 2 months age bird, then we can differentiate it from ALC, because Morix disease generally occurs during the 8 to 17 weeks of age. However, ALC disease is particularly seen in the aged birds, for example, 4 to 5 months of age or above 4 to 5 months of age. So age of birds also plays a role in diagnosing the disease. Similarly, aspergillosis is a disease of young birds, as we all know that. Similarly, IBD and coccidiosis is a disease that is particularly and basically observed during the 2 to 3 weeks to 3 to 6 weeks of age. Similarly, avian encephalomyelitis. So the age of birds also helps in diagnosing the disease. Similarly, clinical signs and symptoms. For example, if there is bloody diarrhea, then it implies that the disease could be the coccidiosis. Similarly, if there is neurological signs, then it could be the sign of avian encephalomyelitis. So and if you see the problem related to the eggs and its quality, then it could be the disease related to IV or AI or ND. So those clinical signs and symptoms also give some basis for the disease diagnosis. Similarly, the mortality patterns. For example, infectious bursal disease has a spiking mortality pattern. The mortality will go high within 1 to 2 days, and it will suddenly decline after 2 to 3 days. So this is the spiking mortality pattern is generally observed in the IBD disease. Similarly, the ALC, avian leukosis complex, is a disease of adult birds, as I have already informed you all. And the mortality is very sporadic. Let's say 1 to 2 mortality in the 1,000 flock per day, or 2 to 3 mortality in a week. So the mortality is very low. So such mortality patterns also give some idea regarding the tentative disease. Okay, the next thing is the postmortem examination. During the postmortem examination, we do the tentative diagnosis of poultry diseases on the basis of typical lesions that we see in the diseases. And also during the postmortem examination, we see the presence of parasitic worms. That means it is, if there is a presence of parasitic worms in the intestine or other parts of the body, then what it implies is the birds has been affected with the parasitic worms. So the postmortem is also a diagnostic facility, and most of the field veterinarians and most of the private or public government laboratories follow the postmortem examination for the poultry disease diagnosis. And the other method is rapid diagnostic test, RDT. And nowadays, the use of rapid diagnostic test has grown up, not only in the veterinary sector but also in the human health sector. So the rapid diagnostic test, RDT, is a a medical diagnostic test which is very quick and easy to perform. RDT is generally used for the screening of the clinical samples or for the surveillance, because it does not quantify, and its specificity is though its specificity is high, around 96 to 97% or even 98%, but it is not acceptable, and the diagnosis is not acceptable internationally. Or let's say if we send send the information regarding the presence of a disease here in Nepal with the basis of RDT, then it will not be accepted by the international organization like OIE. However, the RDT can be used for the preliminary and emergency medical screening purpose. And the rapid diagnostic test it gives result within 10 to 20 minutes. So it is very easy to perform, and competitively it is cheaper also. So how the RDT looks like. Here is the the principle of RDT paired. I would like to share one video through the YouTube so that you will have a better understanding. Okay, so I hope my screen is visible to everyone. It is just 1 minute 52-second video, so have a look. So okay, here what we can see is the different. Yes.
We cannot see your video.
Okay,
YouTube,
okay, sorry.
YouTube video.
Now I'll share screen then. Before, I think it was I was just sharing the the presentation. Now I have shared the screen. Can you see now, Madan? Can you see the screen now?
Yes, we can see, sir.
Okay. So it is not a new thing. It is just available in the YouTube. However, I would like to share this video so that if any one of you have not gone through the video, you can have basic concept on the principle of rapid diagnostic test kit. So this is the test kit which we can see from the outside, and there is such pad inside this kit. So this is a sample pad, means we will drop a sample here in the sample pad, and here you can see 3 test line. However, we have been using just 2 test line rapid test kit. Here is a 2 test line. That means one, for example, if someone some company do have the 2 test line, that means we can diagnose the disease, 2 diseases at the same time. Let's say here test line means avian influenza test kit, and here test line 2 means Newcastle disease test kit. So different kind of sample we can keep here, but in case of poultry disease, we will keep for example, for Newcastle disease and for infectious bronchitis and avian influenza, we will keep a tracheal sample. But for some species, let's say quail and duck, we will take the cloacal swab for the avian influenza. And similarly, for the infectious bursal disease, we will take a sample from bursa and kidney. So the type of sample will differs with the rapid test kit, and it also depends upon the the disease, type of disease. The basic thing is that we need to keep here the virus. So the organ which contains high amount of virus, we will take the sample from that organ. For example, for the ND and in case of ND and AI, the virus is present in high amount in trachea. That's why we will keep the tracheal swab sample here. But in case of IBD, the virus is present in the bursa and kidney. So that's why we'll keep the sample swab from bursa and kidney. So once we keep the sample here in the sample well, it will then get absorbed in the pad that is inside. So here is the sample. We don't know what kind of antigen is present in the swab, but here the test line has a specific antibodies. So here what you can see is these are the antibodies. Let's say the yellow antibodies is of avian influenza, and these red antibodies are specifically for the ND. So the blue, this blue is the antigen. This is a AI antigen, and this sky blue is the ND antigen. So the antibody gets attached to the specific antibodies. Okay, for example, let's say if the test line 2 is negative, then what would have been the video means this red antigen would not have been bind here, so that there will be you will not see the lining in the rapid test kit. But in this case, both antibodies are attached with the specific antigen, and that's why we will see the line over both these test lines. So the last line is the control line. That means whatever be the antigen, it will go and bind with this antibody, so that the control line will be positive. This was the short video regarding the rapid diagnostic test kit. I hope it was quite useful to you all. And you can, if you are still, if you want to learn, you can go to the YouTube and you will get this video over there. Okay. So in Central Veterinary Lab, as I already told you, that we have 4 types of RDT test. One is IVV, it's infectious bronchitis virus. It is IVDV, infectious bursal disease. So in every RDT, you can see the red lining in the control part. So that means the test is valid. The RDT kit is valid. And in the IVDV, in the infectious bursal disease, you can see the 2 linings. This is the test lining, and this is the control lining. So, and NDV and AI, they are also negative. So these are the principal and basic information regarding the rapid diagnostic test kit. And similarly, we also diagnose the disease through the plate agglutination test. And in Central Veterinary Lab, we have a plate agglutination test for 2 organisms. They are Mycoplasma and Salmonella. This is the positive sample for Salmonella. I did this sample before, I think few months ago. So the principle of plate agglutination test is that there will be an antigen antibody complex. So the antigen, you will buy an antigen from the chemical store, and you will withdraw a blood from birds, and you will just separate a serum, and you just mix few drops of serum with the specific antigen. Then it will form an antigen antibody complex. It is simply done in the glass slide, and the result will appear within few minutes. Let's say 1 to 2 minutes. There you can see the precipitation which is due to the antigen antibody complex. So in CVL, we have 2 diagnostic facility, that is Mycoplasma and Salmonella. Here you can see the flow chart of disease diagnosis at CVL. First of all, we will receive the sample, and there will be two types of samples. One is dead birds, and sometimes farmers bring live birds also. So regarding the dead birds, we will conduct a post-mortem, and we will and according to the sign and symptoms and mortality patterns, we will go for RDT test, and depending upon the tentative disease diagnosis, we will take a tissue or a swab sample for the further investigation. And if there is a live birds, we will take a sample. Let's say if we suspect for any viral infection, we will take a cloacal swab or fecal swab for the RDT test. Or if we suspect, let's say Mycoplasma or Salmonella, we'll take a blood and serum sample for the disease diagnosis. And we also can take fecal sample for the parasitological examination. So regarding the dead birds, after we conduct the post-mortem unit, after we conduct a post-mortem, then we will take the sample for the Bacteriology Unit, Histology Unit, Serology Unit, Molecular Unit, Hematology Unit, and Parasitology Unit. In the Bacteriology Unit, we have different methods for the diagnosis of the organism, bacteria. And in the Histology Unit, now it is not a functional unit. That's why I have put asterisk over here, because Histology Unit is now not functional. However, before a few months and years, we used to diagnose diseases, Asian body hepatitis. And in serology section, we do ELISA test and HASI test. And similarly, in the molecular unit, we conduct PCR. And in hematology unit, we do biochemistry analysis. Question may arise in your mind that do we conduct biochemistry analysis for the birds? Yes, we do. For example, a few months ago, we did many samples of pheasant calis, because at that time there was huge problem and there was high mortality in the calis. So at the time we asked farmers to bring the live birds, and we collect the blood samples, and we did biochemistry analysis for the further investigation, because we were unable to diagnose the disease from other laboratory tests. So we did biochemistry analysis also. And in parasitology unit, we do fecal examination for the presence of cystodes or nematodes. And here below, I have kept two condition. If we suspect mycotoxicosis, then we request farmers to send the feed sample to the livestock laboratory, which is in the Hariyar Bhavan, Lalitpur district. And in the lab, they will detect the mycotoxin level through the SPLC equipment. And if we suspect some toxicological samples, if we suspect some malicious poisoning, and in that case, we will send the samples via the content, the feed content in the gizzard or intestine, to the National Forensic Lab, which is in the Sadhu Bato. We send some sample to this lab also for the diagnosis. So these are the units and tests we are currently having at CVL. So here you can see some of the diseases that has been diagnosed at CVL. These are the most common diseases that we are having. Okay, I will now go one by one. The Bacteriology Unit. In the Bacteriology Unit, we have a facility of culture and isolation, gram-fast staining, acid-fast staining, different biochemical tests, and antimicrobial susceptibility testing. So these are the facility for that helps for in the disease diagnosis. So here are some of the automated systems available at CVL. The first one is the BD-Phonics. BD-Phonics is used for the identification of bacteria and also for the antimicrobial susceptibility testing. And there is MALDI-TOF identification. As I already informed you that MALDI-TOF has been installed in our lab in 2021. It was just 1 year ago. And there is another automated system that is a Vitec 2. It is used for the identification and antimicrobial susceptibility testing of the organism. So what is BD Phonics? Here you can see the equipment BD Phonics. BD is in fact it is a company, American company. So Phonics is a instrument equipment. So it is used for the identification and antimicrobial sensitivity testing of the organism. At a time, it can identify 50 organisms at a sample. So what we should do is we have to make a isolation through the culture, and once the organism is isolated, it will be then kept in such columns. First of all, we have to do the gram staining, so that there are two types of column. One is for gram positive columns. One column is for gram positive, and the other is for gram negative bacteria. So before we go for this procedure, we have to identify whether the bacteria is gram negative or positive. Once it is identified, then it is kept in the respective columns. Then these columns are then kept inside this equipment. As I already informed you that this is for the identification, and the pattern of sensitivity, resistance, or intermediate. It also gives the minimum inhibitory concentration level also. So at a time, it gives result of 20 antimicrobial agents, and these antimicrobial agents are already installed inside this column. So the principle of BD Phonics is that it is based on the different biochemical tests. There in the data, in the column, there are different types of chemicals and enzymes kept inside. Then once the organism is kept inside, then it will go under different biochemical process, and it will give the result. And there is the computer system that is installed, and this will give the result. As I already told you, that antimicrobial susceptibility testing, at a time it can identify, it can give result of 20 antimicrobial agents with its MIC value. So this is regarding the BD Phonics, and the other is MALDI-TOF. MALDI-TOF is this machine. So before processing to the MALDI-TOF, it is we need to have isolates, single isolates of the organism, and it is kept in such columns. You can see different spots over here. Then each of these organisms are diluted and kept inside these these wells. This place, in fact, this is not a well. This is just a marked spot. We need to keep those organisms here. Then at a time we can keep 48 samples over here, and there are 4 columns here: 48 samples, 48 samples, 48 samples, and 48 samples. Then these columns are kept inside here through this open through this place, and it is allowed to run for hours. Then it helps for the organism identification. It not only identifies the organisms, but also it gives a serotyping. Let's say, for example, it not only identifies the Salmonella species, but also it identifies Salmonella serovars, for example, Gallinarum or Typhimurium or Paratyphi, Typhi. What kind of serotypes belongs to? It also gives that information also. So this is the output of this MALDI-TOF machine. So the next one is the Vitek. Vitek, it is one equipment which I have already shown here. This is the Vitek machine. It is also connected with the database computer-based system. So Vitek is based. It is also based on the metabolic activities because all of the organisms has their own metabolic activities. So depending upon the metabolic activities, it gives a result. So that is the principle. And regarding the antimicrobial susceptibility test, it gives result within 16 to 18 hours. So at a time, it also gives around 20 antimicrobial agents with its MIC value. And regarding the identification, Vitek 2 can identify the organisms within 4 to 6 hours. So these are the automated systems that is available in the CVL. So these are basically, installed at CVL for the purpose of antimicrobial sensitivity test, because as we all know that, antimicrobial resistance has become a global issue, and nowadays it could be a pandemic in the near future. So this Vitek machine, Veriphonics, and MALDI-TOFs, the automated systems has been installed at CVL basically for the purpose of antimicrobial resistance. So in the Parasitology Unit, we have a facility of Parasitology Unit. Here you can see the oocyst of Eimeria. And similarly, as I have already told you, that we have a Histology Unit, but now it is not functional right now. And there is a molecular unit. We have a PCR, RT-PCR since 2009, as I have already told you. This is a PCR machine, and we use this PCR machine only for 3 poultry diseases. We do for many other diseases of livestock, but regarding the poultry disease, we only do PCR for these 3 diseases. And regarding the avian influenza, we have a primer of S5, S7, and S9. As we all know that there are 16 types of hemagglutinin, and out of those 16, S5 and S7 are regarded as highly pathogenic. That's why we have to do this S5 and S7. And S9 is one of the most prevalent serotype of LPAI. That's why we have a primer of S5, S7, and S9. And similarly, we have a facility for diagnosing the Newcastle disease and infectious bursal disease. Similarly, we have Serology Unit. We do here is the principal. We all are veterinary students or veterinarians. That's why I do not want to go to the principal. However, we have the facility of avian leukocyst complex diagnostic facility through the ELISA. And similarly, we have HAHI test. It is basically done for the AI and MD. And this is the case which I have encountered few weeks ago. This was suspected for malicious poisoning. See here, you can see the crystal of magnesium sulfate. I don't know whether it is magnesium sulfate or not. However, it looks like a crystal of magnesium sulfate. And the farmers, brought the dead birds suspecting the malicious poisoning. And the farmer has not given the birds with the meat, but we see here the pieces of meat and bones. So we suspect and we refer that to the National Forensic Science Laboratory. So regarding the reliability of laboratories that we are having here in Nepal, as I already told you that CVL is the National Reference Laboratory and recognized by the OIE. And we do regular proficiency testing. So proficiency testing is one of the quality control. International reference laboratories send us the sample, and we conduct the sample, and we send those the results of the sample. Then they will, and this is called the proficiency testing. And this is generally done for the quality of the lab. And our lab is not still accredited. Because the laboratory, the ISO 17025 is a accreditatory body for the laboratory. So till date, we do not have any laboratory organization, even in the private or in the government organization that is accredited. And similarly, we are the member of International Atomic Energy Agency. Till now, there are 173 member countries of IAEA, and we are among one of them. So the future of diagnostic facility. So here are some of the future of diagnostic facility in the poultry sector. Till now, we do not have the facility of gene sequencing. So Central Veterinary Lab do have the equipments for the gene sequencing, but due to lack of human resource, skilled human resource, we are not being able to do the gene sequencing. However, there is a private sector, and its name is BioVac. I think BioVac has been doing the gene sequencing of the organism. And similarly, RDT and PAT at field levels, now RDT and PAT are only available in some of the veterinary clinics, but it's not available throughout the country. So in the near future, it is very important to have those tests at the field levels for the disease screening and for the clinical disease diagnostics. And similarly, as I already informed, the automated systems at the CVL, it is still not being used for the clinical diagnosis, because clinical diagnosis, because the number of clinical samples in the CVL is, let's say, 10 samples per day, and every samples do not require the bacteriological identification. So due to the less number of samples, we are not being able to run those automated equipments for the clinical samples. However, it is being used for the surveillance sample. So we need to use those automated diagnostic equipment for the clinical samples also. Similarly, molecular diagnostic capability is only at the CVL for the clinical diagnosis. It is not available as a clinical diagnosis in the veterinary laboratories and other private laboratories. So it should be, it should be installed, and it should be bring in use in other laboratories also. And we do not have our own veterinary forensic laboratory, so it is must. So I think in the near future, we will have our own forensic veterinary laboratory. And as I already told you that our lab is not accredited, so that's why it is needed, it is required. And similarly, we do not have a diagnostic facility of new poultry diseases. For example, poultry practicing vets has been encountered few cases of chicken infectious enema, infectious laryngotracheitis, even encephalomyelitis or other adenovirus such as egg drop syndrome, or let's say intrusion body hepatitis or leachy heart disease. So we do not have the diagnostic capability of such new poultry diseases. So we need to strengthen those capacity in our veterinary laboratories. So these are the future of diagnostic facility, and this is all about my presentation. For now, thank you so much for being with me. I think I don't know how much time I spent. So if you have any queries, please let me know through chat box or through audio. I'll be happy to receive the queries.
Thank you so much, sir, for your wonderful, informative, and practical-based presentation. If you have any queries, questions regarding the topic or presentation, you can ask directly by unmuting yourself, or you can drop in the chat box.
Okay, there is one question from Binay Thapa. His query is regarding why only 3 diseases diagnosed with PCR. There are a few reasons behind this, because PCR itself is a quite expensive test, because the primers, the cost of primers is quite expensive. We have primers of various diseases. I think more than around 20 diseases, but they belongs to the cattle, horse, like glanders we have. So there are about 20 primers we have, but regarding the poultry disease, we have only 3. And we are planning to have primers of other diseases such as Morex disease and ALC also. ALC, I think we are planning to have these 2 primers in the coming year. So the reason is that basically the number of cases of other diseases is quite low, and the primers is quite expensive. That's why we only have the primers of the diseases that is frequently observed in Nepal. So this is the one reason, Vinay. Okay, thank you so much. Okay, there is one more question from Dr. Vivek Regmi. Do the samples which we use in rapid diagnostic test differ sometimes as per the manufacturing companies? I'm not sure whether it differs or not. However, you also know that the basic principle is that the sample needs to have high amount of virus. So what we generally do in the lab is we, though manufacturing companies, specify the specific sample, we take the samples from that specific organ, and also we will mix those sample from the other tissues also sometimes. For example, regarding the infectious bronchitis, we all know that infectious bronchitis not only affects the respiratory system, it also affects the kidneys. That's why in this case we take the samples from the trachea also, and we also take the samples from the kidney. So sometimes we can do by our own knowledge. And there is high risk of getting false positive samples in case of rapid diagnostic test kit, because sometimes what happens, let's say if we are doing test for the IVD or let's say ND, sometimes there is chance of getting false positive samples, because if the birds are vaccinated just 2 to 3 days ago, and the live vaccines that is present in the trachea or in the bursa may affect the sample to be positive. Say about biosafety level BSL2 and 2+, and why other not effective in Nepal. Okay, the biosafety level laboratories are classified generally on the basis of the equipments or facilities that is available in the in the laboratory. For example, BSL lab 1 means the basic laboratory, and BSL lab 2 means the facilities which is better than BSL 1 but less than BSL 3. In BSL 3, there is some facility. Let's say there should be a double door system. There should be a negative pressure at the door, and there should be a facility of biosafety cabinet. However, in the BSL 2 lab, in the BSL 1 lab, there is the biosafety level cabinet or laminar flow is not required. However, in the biosafety level 2, biosafety level 2, there should be a provision and facility of biosafety cabinet, laminar flow, and in biosafety level 3, we have there should be a capacity of double door system and HEPA filters. In case of our CVL, we have this facility. However, it does not, it does not meets the every standard that should be present in the biosafety level 3. And there are no any biosafety level 4 in Nepal. So I hope you got the answer, because biosafety level 3 or 4 is it's a highly sophisticated and highly equipped lab. So till now we do not have such budget to go for biosafety level 3 and 4. However, there should be I hope government will soon allocate some budgets. Why is the histology unit in CVL not functional now, if it can be shared? I think the main reason is that we do not have, the expert human resource at CVL, because we all know that in government sector there is, high mortality, because if someone is working in the histology lab, he will be transferred in the next month in the other laboratory. So due to this reason, due to this, high chance of having transfer from one office to another office. So, and we all know that histology requires quite expert skill. So this is one of the main reason. Otherwise, there is no any issues with the equipments. The only reason regarding the histology unit is we do not have skilled manpower to work in the histology unit. Is there any more queries? Okay, in fact, today's lecture is just, it's a practical based question and just an informative ones. This is not a, I hope this, I know that this presentation will not upgrade your knowledge, your, technical knowledge. However, it gives information regarding the diagnostic capability and capacity in Nepal, because we are having, the participant from the universities who are studying in, let's say, first or second year, third year. So they might not know the capacity for the disease diagnosis. So this presentation has just give the idea and the knowledge on the diagnostic capability that we are having in Nepal. So as I already informed you in the limitation of this presentation, that we do not have, this presentation does not, upgrade your knowledge regarding the procedure and other things. So I think if there is no more queries, we can end this presentation. Madanji.
Okay, sir. Thank you for your presentation. And we expect your support towards IVSA RAMPUR as always.
Okay, definitely. I would also like to, give my huge thanks to the IVSA RAMPUR for organizing such type of technical seminars, and I'm very much happy to be a part of it. And I would also like to I would like to give thanks to all the participants, national and international participants, for being with us since more than one hours. So thank you so much. Hope to see you again. Thank you, Madan.
Thank you so much, sir.
Okay, thank you.
And thank you so much all the participants for their active participation. And finally, now I want to invite Mr. Paramod Kumar Chaudhary, Mr. President, for his concluding speech. Thank you.
Thank you, Mr. Madan Kanal, the host of the program. Regarding the program, the session was very informative and interesting, and we have got a very good number of very enthusiastic participation, and we also get participation from the international level, which is very good for us. And we got the chance to know a lot of information about the poultry disease diagnostic techniques in our country, and we also get insights of its future use. So I would like to thank Dr. Nabaraj Shrestha, sir, for the speaker of today's program, for such a wonderful speech. And it was very generous of you to manage time despite being your busy schedule for the program. And lastly, IVSA RAMPUR would like to thank all the participation for making your valuable presence and making the program successful. Hope you have learned something new from this program. By saying this much, I would like to officially announce the end of today's session. Thank you. Stay safe and stay connected.